Today our topic is spirochaetes. Let us see the most common points of spirochaetes. They are gram-negative 1. They are the motile organism. They are tightly coiled bacteria, typically slender and flexus shaped. The cell made up of the protoplasmic cylinder located in the periplasm of the cell, interflagella and the outer sheath. They move by bending, rotating the body according to the viscosity of the medium. While speaking about the motility of the spirochaetes, they are unusual mode of the motility performance. The endoflagellum located at the one end and extends about 2/3 of the cell. It's rotate. The protoplasmic cylinder is rigid, whereas the outer sheath is flexible. Therefore, when both endoflagella rotates the same direction, while the protoplasmic cylinder in the opposite direction, causing the torsion of a cell. Medical important. There are 3 genera. One is Treponema, Leptospira and the Borrelia. Treponema. The Treponema pallidum, which causes the syphilis. Treponema pallidum endomysium, which causes the endemic syphilis. Pallidum keratium, which causes the pinta. Pallidum peteneum, this causes the yaws. These are all the common diseases caused by the Treponema. Normal habitat. They are host-associated spirochaetes. They are the parasites of the human or the animals. They are found in the lesions of the Treponematous. Can also found in the mucous membrane in the mouth and genital tract, also in the skin also. Pathogenicity. Acquired syphilis, transmitted congenitally or the sexual intercourse, known as the sexually acquired syphilis and congenitally acquired syphilis. Non-visceral syphilis, endemic syphilis, Bijan, Nogueira, CT and the Zichovia. Microscopy, small 6 to 15 mm size, 0.2 mm diameter. Motile spirochaetes. They use the metallic silver techniques are known as the impregnation techniques. Metallic silver precipitates on the wall of the spirochaete, enlarging in outline and making it opaque, features that make it visible in the tissue sections. They are best seen by the dark field illumination or the phase contrast. While speaking about the culture of the spirochaetes, microaerophilic, hardly to grow. In the routine culture, able to survive in some fluid including the cicatricial whole blade. Maintained by the intratesticular inoculation in rabbit. Serology test, cardiolipin antigen test, VD oral test. Venereal diseases reference laboratory. RPI test, rapid plasma reagent, treponemal antigen test. TPHA test, Treponema pallidum hemoagglutination test. Leptospira. They are the scanning electron microscope of the number of the Leptospira species bacteria at top of 0.1 mm polycarbonate filter. Normal habitat. They are gram-negative, obligate aerobe spirochaete, variety in domestic and the wild animals such as rats, rodents, cattle, pig and dog. Humans are the accidental host here. They are largely secreted in the urine. They survive many weeks in soil and water in alkaline and they will be at the temperature of 28 to 32 degrees Celsius. While speaking about the transmission, direct or the indirect contact with the urine of the infected animal enter the damaged skin which has immersed for the long time in water or mud contaminated with the infected urine. They penetrate in the lining of the mouth, nose and eyes. Person-to-person transmission rarely occurs. The risk factors like the Leptospira will be in the urine. They may be present as in the contaminated water, mud and soil. You can get the disease using when you are planting, hunting, getting, weeding, skinning, fishing, bathing, harvesting, climbing and swimming. Risk factors like the people most at the risk of infection include the workers in the rice field and the sugarcane field, livestock farmers, irrigation canal workers, meat and the animal handlers, freshwater fish pond workers, veterinarians, pest control officer, forest worker and those involved in the road building. High cases in the rainy season and times of flooding. While speaking about the pathogenicity, mild form usually misdiagnosed as the viral illness and the influenza. Most serious infection causing the high continuous fever, severe headaches and the body pain, especially in the muscles of legs, redness of the eye and weakness, can also causing the jaundice and the renal failure. Now we can see the microscopy. They are small 6 to 20 mm in size, 0.1 mm in diameter. They are actually actively motile spirochetes, screw tight coils and hooked ends. They are easily stained only by the impregnation techniques, best seen by the dark file elimination or the phase contrast one. Culture. The leptospires are strictly aerobics. They can be cultured in the serum or the albumin twin medium. They are let grow, so they are resulting in the less efficient diagnostic, preferable serology test to identify the several serogroups. They test the hematological and the biochemical test in the leptospirosis, antimicrobial sensitivity. High doses of the penicillin will be effective against the L interrogans. ELISA test polymerase chain reaction. Borrelia. The normal habitat is Borrelia burgdorferi. They will be in the dead tick. Recurrentis. They are the parasite of the pediculus lice. Deutony. They are the parasite of the Ornithoderus soft ticks. Pathogenicity. The Borrelia burgdorferi causes the Lyme disease. Recurrentis, they causes the louse, burn, relapsing fever in the poverty, overcrowding and during the time of drought and famine. Deutony, they are the tick-borne relapsing fever infecting the CSF causing the lymphocytic meningitis. While speaking about the microscopy, they are large, 10 to 20 mm in size, 0.5 mm in diameter, actively motile spirochetes with the coils of varying size, agglutinate together, easily stained in the Giemsa, fields or other Romanovsky strain, prolonged gram staining, well seen by the dark field elimination. The culture of the Borrelia or the aerobes, they can be cultured in the caelus medium. Serology tests, they are used to diagnose the relapsing fever, usually cross-section with the Treponema. They are antimicrobial sensitivity. They are sensitive to penicillin and the tetracycline. Thank you.